Thanks for joining in again, guys. This is Mr. Dundon, and today's tutorial will be all about M8 of the Stage 2 Biology curriculum. M8 is all about enzymes, and more specifically M8.1 is for students to be able to describe the induced fit model of enzyme-substrate binding. Now, before we dive straight into enzymes, it's probably a good idea just to do a quick little review. So we know so far through the course that we have a DNA molecule, and that can be transcribed into several things. We can get RNA, making ribosomes, and they're actually going to be involved in protein synthesis. We can get tRNA, and we know they're involved in carrying specific amino acids, and the tRNA itself has an anti-codon that codes to our, which is complementary to our codon on an mRNA. So we also have mRNA, and that mRNA is transcribed from DNA, from a gene which is complementary, and that mRNA will then go on to code a polypeptide. And that polypeptide is then going to undergo its structure, secondary structure, tertiary structure, and possibly a quaternary structure, and then that will lead to a protein. So really this protein is governed by its primary structure, its amino acid sequence, which is governed by the mRNA sequence that coded for it, which is governed by the DNA sequence on the template strand that coded for that initial mRNA. So how does this relate to enzymes? Well, quite easily, or quite simply, enzymes are proteins. So therefore we get different enzymes by transcribing different genes, different mRNA, different polypeptides, producing different proteins. Therefore we can have an endless number of different proteins, therefore different enzymes. And that's very important because enzymes are catalysts within the cells. So a catalyst is something that alters the reaction rate of reactions, either speeding it up or slowing it down or controlling how fast it goes. So that's really important. So they're our catalysts. So therefore if we want to be able to have a certain level of control, we need to have intermediate steps. And therefore we can do that by having different enzymes control different steps of a reaction. Okay, so now we're going to go into exactly how this relates to our induced fit model, and so let's dive into that now. So this particular tutorial will be divided into two parts. The first part is how do we have this sort of enzyme controlling reactions, and then what affects these enzymes. Okay, so the induced fit model is very simple. We have an enzyme that has a specific shape governed by its protein structure. So let's just remind ourselves, we have our primary structure, and that is just the sequence of amino acids. They're amino acids, by the way, and they've got their peptide bond, and because there's more than a few, it's a polypeptide chain. We then have the secondary structure, which could contain alpha helices or beta pleated sheets, and they're caused by hydrogen bonding. So just remember hydrogen bonding for that secondary structure. And then we have our tertiary structure, which is really governed by the R groups on each amino acid. So remember, an amino acid has an amine group, a carboxyl group, and an R group. That R group is different between the 20 amino acids, and that gives it some characteristics. So the main thing that we're going to see is some basically hydrophobic, hydrophilic interactions because of the polar nature of some of the R groups. So we'll see some, we'll just put that for H, hydrophobic, hydrophilic interactions, polar interactions. We see some acid sort of interactions, ionic bonding. We see some disulfide bonding. We see some hydrogen bonding, put that disulfide bonding, and we can see some van der Waals interactions as well. So all of those will give its three-dimensional shape. Okay, and that's going to be different depending on what this amino acid sequence is, which then goes back to our mRNA chain or our mRNA sequence, which goes back to our DNA sequence. So you can see how they're all really interrelated. So we've got our tertiary, our three-dimensional shape. We then might have a quaternary structure if we've got more than one polypeptide joining together. So enzymes are big proteins. Okay, so the induced fit model. First of all, let's just draw an enzyme. I'm going to draw it as a diagram. So this particular enzyme has this shape, okay, and it has a three-dimensional shape governed by the amino acids and all the interactions forming that tertiary structure, and we're going to have this open area. This open area is what we call the active site. So that's our active site. Okay, so we've got our active site, and the active site is specific. It's specific to that particular enzyme. Now, we also have, so this enzyme is going to govern a reaction, so we need to have something that's doing some reacting. So we call those substrates. So this substrate that I'm going to draw, I'll draw it in red, has this particular shape, very interesting shape, and it will fit into our enzyme active site. Okay, so now we can basically draw that in there. Okay, so it can go into our active site, and the enzyme is going to catalyze a reaction, and we're going to call this A, and what comes out is going to be a product B as well as a product C. So A goes into our active site, the enzyme catalyzes a reaction, and it's going to split A up into B and C. Okay, very simple reaction, and one that this enzyme is going to do over and over and over and again. Because it's very important to remember that although the substrate, so although the substrate has been altered, the enzyme itself remains unchanged. So we can go over and on and on and on and over and over and over again doing that particular action, making A into B and C, making A into B and C, making A into B and C over and over and over again without changing. Okay, so that's very important. So we call these our products. Okay, I'm drawing with my finger, by the way, so it's quite difficult. So substrate A goes into our enzyme, and then a reaction occurs and forms B and C. Now the particular M8.1 said the induced fit model. Okay, so the induced fit model is once the substrate binds to the active site, so a specific substrate is complementary to the specific active site of our enzyme, once they bind, we get a conformational change. So we get a conformational change to that enzyme active site, and it gets a little bit tighter. So it just comes in a little bit, gets a little bit tighter, some rapid changes occur, it gets a little bit tighter, and that actually facilitates the reaction. So it makes it go better. So that's all the induced fit model means, but there's still some key things that you need to remember. We have a substrate, complementary to our active site. The active site of the enzyme is then able to catalyze that reaction, producing B and C. And that's basically all you have to remember. Okay, so if we want to control the breakdown of A, we need a number of enzymes that is going to be complementary to A. So in this case we've got this enzyme, and this enzyme is going to have a specific active site that meets the particular three-dimensional shape of that substrate. It can then catalyze the reaction, forming products. It might be two substrates and one product. It might be one substrate and two products, or it might be any combination that causes that particular substrate needs. So I hope that's very clear. Once the substrate binds, we call this the substrate enzyme complex, and then during the change, so during the reaction, we call it the substrate product complex, and then we have our products. Okay, so how does that enzyme actually catalyze the reaction? What is it actually doing? Well, what it's doing is it's making sure that the substrate, if there's more than one, are going to be in the correct orientation. Okay, so they're going to be in the correct orientation. It is then going to perform stress, or provide stress on the bonds of those molecules, making new bonds. So when we have new bonds, we have new products. And by being in that sort of tight, basically the tight conformational change, the tight induced fit model between the substrate and the enzyme, we're basically lowering the energy that's required for that reaction to take place. Okay, so key bits of information. The substrate is specific to the enzyme. Okay, enzyme is specific to the substrate, and so if we want a different substrate reaction, we have to have a different enzyme, so therefore we have a different amino acid chain from a different mRNA from different DNA. Okay, and that's basically it. If you have any questions, please ask them below. Make sure you subscribe, give me a thumbs up, and look forward to new videos. Thanks for your time.